All right, so today's notes are going to be on cellular respiration. So we've already talked about photosynthesis for a while now. So we're going to talk about the opposite process, technically, which is cellular respiration. And I want to put out a little reminder that cellular respiration is a process that all living organisms do, including plants. So some people have initially the misconception that plants only do photosynthesis and then heterotrophs only do cellular respiration. And that's not completely true because plants can do both, right? They're storing their energy, but they're also going to need to use energy in order to, survive and do metabolism. And then on the other hand, there actually have been these new sea slugs that have been discovered that have green little parts inside of their cells that actually do something similar to photosynthesis. And that's kind of cool because science is an ever evolving field. And when you make new discoveries like that, you realize that organisms are adapting always to their environments. And in some cases, there's not maybe the best food source for them to go and find, so they can end up making their own, or they'll do something close to like the endosymbiotic theory and absorb bacteria that can do photosynthesis and make it a part of their internal ecosystem. So I think that's pretty cool. So that means that not animals aren't only just doing cellular respiration anymore, because some of them are turning out to also be starting doing photosynthesis. Okay, so our do now right now is actually just kind of a pre-quiz. I don't know if you know all of these terms, and I don't know if you know how to answer all of these questions. However, I would love to see if you could try. So in your notes, the do now section, I do want you to write down these questions and then see if you can answer them. And if you don't, that's okay, just leave them blank. I just want to, like, as I'm previewing your notes for your extra credit, to kind of see where we are starting from. Okay, so first one is list the inputs of glycolysis. If you're not sure what that is, you can look it up, but I'll also go over it here. List the outputs. Where does glycolysis occur? List the inputs of the citric acid or Krebs cycle. Those terms are used interchangeably. So when I say citric acid cycle or I say Krebs cycle, they mean the same thing. And as you can already kind of tell, cellular respiration is similar to photosynthesis, where it's broken down into two main parts, two halves of the reaction. There's a bunch of little steps that it's broken further down into, and we'll actually go into those details, but obviously you can see glycolysis, and then you'll see the citric acid cycle. If you know, tell me the outputs of the citric acid cycle. This would be the final output. So if you know what the final output of cellular respiration is, you should be able to answer number 5. Likewise for number 1, if you know what the inputs are for cellular respiration, you should be able to answer number 1. And then number 6 is, where does the citric acid cycle occur? And I've drawn like a kind of weird looking diagram of a mitochondria, and you kind of get the idea where it's like a bean-shaped organelle, and inside the membrane is very, very folded. And that folded inside membrane is called the cristae. And then between the two membranes of the outside membrane and the folded membrane, there's this intermembrane space. It's literally just called that. It's not a very creative name, but it is, exactly what it says. There's an outer membrane, and then the space inside of the inside folded membrane is called the matrix. Okay, it's not related to the movie, it's just what it's called. Okay, so we're going over cellular respiration notes. Again, your chapter 8 summary are due on Friday at midnight, not Sunday, because obviously break is starting. And for the learning objective, we are going to be talking about the use of free energy today in cellular respiration, because photosynthesis and chemiosmosis was related to capturing and storing energy, but now we're going to be talking about breaking the storage molecules and using the energy that were originally stored. So in cellular respiration, this is related to what we talked about before with the vocab that's related to chemistry. Remember at the bottom, I wrote again here that oxidation is removing and losing electrons, and reduction is gaining or adding electrons. So when we talk about the carbon and the oxygen molecules, we're going to be talking about them gaining or losing electrons. And that's how they change into a new molecule and how they change from their reactant to their product. So in cellular respiration, you're going from glucose, where the carbon is, to carbon dioxide. And in that process, you are losing electrons from the carbon molecule, from the carbon element or the carbon chemicals. And as they lose electrons, they're going to be bonded differently to different chemicals, and therefore you're losing those bonds from the H's and the oxygens directly on the carbons, and it's going to end up just being carbons bonded with oxygen, and that's oxidation. And over here we have the oxygens getting reduced, and when they're reduced, you're adding electrons, and when you add electrons, they need to make different kinds of bonds, and therefore the hydrogen is coming in. The focus of cellular respiration isn't actually on the oxidation and reduction of the chemicals, though. It's actually on the energy that is produced, because the whole point of doing cellular respiration is to get usable energy that you can use in further reactions to power other parts of metabolism. So we're going to be mainly focusing on the energy that's produced as the molecule known as ATP, or adenosine triphosphate. So like I said before, there are two main parts of cellular respiration, but they're broken down into 4 phases. So the first main phase is glycolysis, and we're going to go into more details with diagrams in the following slides, but I kind of want to give an overview right now. So if you want to wait until the end to come back to this part of the video and write this all down as a summary, that's also a really good idea. So glycolysis is the breakdown of glucose. So you have the initial 6-carbon molecule, which is glucose, right? It's a ring with 6 carbons in it. But we're breaking that down into individual carbon dioxides, which means that you have to rip off carbon at a time. Okay, so you're going to break down that 6-carbon glucose into 2 molecules of pyruvate. And that means you're taking this 6-molecule glucose, 6-carbon glucose into 2 3-carbon pyruvates. And once they're broken into this 2 sets of 3, they're going to move into the next part. So this part of the cellular respiration process actually occurs in the cytoplasm. That means that organisms that do not have mitochondria can still do glycolysis. That's like bacteria, archaea, they can still do glycolysis. So that is a unifying process throughout all living organisms, whether or not they have membrane-bound organelles. And that's really cool because it kind of shows common ancestry. Okay, so there's a little bit of ATP formed. There are 2 molecules of ATP formed during glycolysis. And honestly, during cellular respiration, we talk about net gain, right? So when we write over here in the formula where there's energy produced, that's because we have a net positive gain. But the 2 created in glycolysis, if you're not doing the rest of cellular respiration, yeah, that's your net gain, right? You get a +2 of ATP. It's not a lot though, right? The whole point of cellular respiration is to make a lot of ATP. We're talking in the 30s, close to the 40s, right? But if you are going to go from glycolysis into the rest of cellular respiration, these 2 ATP actually get used up. Okay, so this part of glycolysis does not use oxygen. So if an organism is in an environment where there is no oxygen or it is starting to lack oxygen, then glycolysis is the only part of cellular respiration that you can actually perform in your cells. And there are some bacteria that live where there's no oxygen, and that's why they can only do glycolysis, because they don't have the membrane-bound organelles and there's no oxygen available for them to break down glucose with. So glycolysis happens in organisms that have mitochondria and don't have mitochondria. So the next reaction is like a small substep between the main reactions of glycolysis and the citric acid cycle. And this is called the prep reaction. So the pyruvates are oxidized, aka they are losing electrons after they enter the mitochondria. And the prep reaction is like kind of if you're going from the cytoplasm and you go and enter into the mitochondria, this is when it's happening. So the electron energy is stored as an NADH. So you get NAD plus and you're turning it into NADH, and that energy creates this electron carrier that's going to go to the electron transport chain. So we mentioned the electron transport chain when we talked about photosynthesis. There's also an electron transport chain when you're talking about cellular respiration. And it's similarly set up because you're creating that gradient of hydrogen ions, the protons, and then you're going to move it across the membrane through ATP synthase to make a lot of ATP. However, the carrier is different, okay? So in cellular respiration, NAD plus and NADH are the carriers. In photosynthesis, it's NADP plus and NADPH. So an easy way to remember that is when you see it and the carrier has a P in it, that means it's P for photosynthesis. Okay, no P, it means that the carrier is for cellular respiration. I know it's confusing because the rest of the letters are the same, but just use that as your one hint. When there's NADPH, it's for photosynthesis. And when it's NADH, it's for cellular respiration. Okay, so during this process, you originally started with the 2 3-carbon pyruvates in the prep reaction, and then you're going to rip off 1 carbon from each of those, and you're going to end up with 2 carbons molecules going into the citric acid cycle. And those carbons that got ripped off from the third of each of them are going to be released as carbon dioxide. So you're already releasing 2 molecules of carbon dioxide as you're going into the mitochondria. So you actually haven't even done the citric acid cycle yet, and you've already lost 2 of the carbons, which is a good thing, right? So we're already going through and you're already producing some of the products of cellular respiration. Now, in the citric acid cycle, it occurs in the matrix of the mitochondrion. So that means that you're actually producing the rest of the products using the matrix right here. So inside of this space, inside of the sorry, my phone was vibrating. Inside of the space, inside of the inner folded membrane, that's where the rest of this is occurring. So it occurs in the matrix of the mitochondrion. You're producing more NADH and more FADH2. And FADH2 and NADH are both just electron carriers. That's it. They're going to go to the electron transport chain, and that's their sole purpose. They drop off their electron, and they come back to receive more. Okay, you don't need to know any other details about them other than that. Okay. The 4 carbons that originally came in as the two carbon molecules are going to be ripped apart each and turned into 4 separate carbon dioxide molecules. So you've finished producing your carbon products, in this case, which are the carbon dioxides. Okay, so each time there's a molecule, you can only rip off 1 carbon at a time going through the cycle. So you're going to go through once and rip off a carbon, and then go through a second, and then process that second carbon. You're going to rip off 1 carbon, and then you're going to go through again and rip off that second one and process it. Okay, so it's 2 times per glucose molecule, just because you can't break down. Sorry, let me clarify that. It's 2 times per glucose molecule because you're coming in with the glucose molecule, and originally you have 6. Right? And then you're breaking into 3 and 3, and then you're breaking into 2 and 2. And so you go through the cycle once for this one and once for this one, because you're breaking off one of the carbons and then the other one is freed automatically, right? Because it's the only one left. And then you're breaking off one of the carbons and then the other one is free automatically. So you're going to also produce 2 ATP molecules, 2 net ATP molecules during this process. The last part is where most of the ATP is created. And this electron transport chain, the whole purpose of it is to create a lot of ATP. And you're using the electron carriers from earlier that we were talking about, where you have the NADH created during the prep reaction, and you have the NADH and the FADH2 created during the citric acid cycle. So the energy is extracted from these molecules now. The electrons are shuttled, they've been brought over, and they're going to go down the electron transport chain, and they'll lose energy as they're going. But as they're going through the electron transport chain, the proteins along the membrane, they'll create a concentration gradient, pushing a bunch of hydrogen ions to one side, and they're going to push it into the intermembrane space, and that's this space right here in between the 2 membranes. So outside of the cristae, outside of the folds, but inside the actual external membrane to the rest of the system. So this concentration is going to then allow you to have the concentration flow along the concentration gradient through ATP synthase. And so much flows through that ATP synthase will produce 32 to 34 molecules of ATP. Right? That's a lot all of a sudden compared to the 2 that's only producing glycolysis. So as a net, you're producing around 2 up here, you're producing around 2 here, and then you're producing 32 to 34 down here, which means that your total ATP production can be 36 to 38. And it's not exactly consistent every single time because it's based on a gradient and not specific numbers one-to-one matching. So sometimes you produce under 36, you'll produce like 30 to 32, and you can also create upwards to 40 ATP at a time, just going through the cycle with 1 molecule of glucose. So in specific detail, you can choose to write down the bullets, or you can choose to write down in the sentence format. So again, glycolysis is occurring before you actually go into the mitochondria. So it's occurring in the cytoplasm. That's why it's shown out here, and the mitochondria right here I've blocked off with a word box because you're not using it yet. So during glycolysis, glucose gets broken down into 2 pyruvates in the cytoplasm. NADH and 2 ATP are also produced. Pyruvate will go into the prep reaction before it actually goes into the mitochondria, and NADH will be used to the in the ETC, or the electron transport chain, so it'll be moved over there. ATP will be used for energy, which is its purpose as an energy molecule. In the prep reaction, pyruvate molecules get oxidized as they enter the mitochondria for the citric acid or Krebs cycle. More NADH is made for the electron transport chain as an electron shuttle, and then 2 molecules of carbon dioxide are released because you're breaking those 2 pyruvates and releasing 1 carbon dioxide from here and 1 carbon dioxide from here. So you have two carbon molecules entering the citric acid. In the matrix for the citric acid cycle or the Krebs cycle, acetyl-CoA, which is an enzyme, right? We know rubisco from photosynthesis. We also have acetyl-CoA in cellular respiration. So acetyl-CoA, NAD plus, and FAD enter. Right? These are all the reactants for the Krebs cycle. And then NADH, FADH2 are produced and sent to the electron transport chain in order to help pump hydrogen ions into the intermembrane space to make that gradient. And then 4 molecules of carbon dioxide are released. This part of the process also produces 2 little ATPs, but it's not a lot. And then lastly, the electron transport chain, where all of our ATP are basically being produced. So on the cristae, so on that folded membrane on the inside, NADH and FADH2 drop off energized electrons that travel down many proteins, and they lose energy as they're doing so. But when they're losing their energy, they're using that energy the proteins are using that energy to pump the hydrogen ions, or the protons, into that intermembrane space. Between the membrane of the folded cristae and the outer membrane, space in between is completely filled with hydrogen ions, which means it's actually really, really acidic because of all those protons. So with that concentration gradient, then the only way that the hydrogen ions can leave to kind of balance it out into trying to even out the concentrations would be through ATP synthase. And ATP synthase, then as something is moving through it, the ions are moving through it, is going to turn a rotor basically and produce ATP from ADP and a phosphate. So after each single glucose molecule is used up, you can produce 32 to 34 ATP for a total, when you sum up from glycolysis and from the prep reaction and from the Krebs cycle, to end up being around 36 to 38 ATP. So we know that cellular respiration, for the main part that produces a lot of the ATP, requires oxygen. And oxygen only became common on Earth about 2.7 billion years ago. So that's actually not that, like, oxygen didn't exist on Earth for a large period of time that the Earth actually existed. So what organisms increased when the oxygen concentration also increased? It was the cyanobacteria, or blue-green algae. What advantage did oxygen provide to organisms that could use it? Well, when you have oxygen, you can do the Krebs cycle, or the citric acid cycle, so you can produce a lot more ATP. More ATP means faster or more metabolic reactions. So that if you can do metabolism better and you can do it more, that means the organisms can be bigger and more complex and have more processes going on at the same time. So without oxygen being present on the Earth, organisms would never have developed the current size that they are now. And there might have been a point where oxygen was even more abundant in the atmosphere, because we used to have really, really large organisms like the Brachiosaurus or Tyrannosaurus rex, right? You have to have a lot of ATP in order to supply all of those cells with energy. So maybe at some point there was a lot more oxygen present in Earth's atmosphere than there is now. This is kind of reiterating earlier when I was saying the energy carriers. NAD plus turns into NADH. Okay, here's the actual breakdown of what the NAD stands for. So it's nicotinamide adenine dinucleotide. So nicotinamide adenine dinucleotide. There's a reason why we just call it NAD plus. Okay, and when you add the hydrogen to it with the energized electron, and it's going to carry over to the ATC, that's like literally its only job, is to carry that energized molecule. So it gets recycled because it's not going to be destroyed. It's not going to be changed in any way. So it's just going to drop off that electron. The hydrogen ion gets dropped off too, because that's going to be the proton that gets pumped into the intermembrane space. And then the NAD is going to return back up to either the prep, or it's going to return up to the Krebs cycle. I made another note here not to mix it up with NADPH, because remember, NADPH or NAD plus are both associated with photosynthesis, right? Same job though, with different molecules. So FAD is flavin adenine dinucleotide, which is another coenzyme which does the same thing as NAD+. It's just a secondary receptor, and it's a secondary receptor because the bonds it makes, it has to bond with 2 electrons at a time and 2 hydrogen ions at a time to make FADH2, because it's a doubly charged molecule. So it's not as easy for it to make both of those bonds and then to go and drop off both of them, but that means you're doing double the duty or double the work of carrying it over. So FADH2 isn't used in any of the other reactions except for in the citric acid cycle. And again, it's going to be carrying the electron over as FADH2, dropping them off, dropping the hydrogen ions off to be pumped into the intermembrane space, and then it's going to return back to the citric acid cycle to then get re-energized and get another electron added to it. So here is an overview if you need to write down the steps of the citric acid cycle in a little bit more detail if you want it. So in step 1, it says the cycle begins when a carbon-2 acetyl group carried by CoA combines with the C4 molecule to form citrate. And you don't have to know these details, I'm just going over it. Okay, so twice over, it says 2 times over, substrates are oxidized as NAD plus is reduced to NADH and carbon dioxide is released. So it's going twice over because each time it rips off a carbon, that carbon turns into carbon dioxide when it gets, I think it was oxidized. And then NADH is getting reduced so that hydrogen and that electron is getting added to it, right? You have to, when you take an electron from something, you have to give it to something else. So here, when you take the electron away from the carbon, you're giving it to the NAD plus and turning it into NADH. So step 3, ATP is produced as an energized phosphate is transferred from a substrate to an ADP. So this is actually the point in the Krebs cycle where the ATP is being created. And again, remember, we're going through it twice. So the second time is the second ATP. That's why it produces 2 ATP overall from 1 glucose. So in step 4, again, a substrate is oxidized, but this time it's FAD, reduced to FADH2. Okay, so this is where the second carbon is going to be oxidized, and then FAD is reduced to become FADH2 as the electrons are getting added to it. And then once again, the substrate is oxidized and NAD plus is reduced to NADH, and you're starting the cycle over again to do the other molecule of the 2 carbon molecule that came from the prep reaction. So if you could, I want you to pause and I want you to just summarize the steps of cellular respiration. I want you to start with glycolysis and then finish with the electron transport chain. I just want inputs and outputs, purpose, and where. So they should be sentences. You should be able to answer this in 4 sentences because there are 4 phases, right? You have glycolysis, you have the prep reaction, you have the citric acid cycle, and then you have the electron transport chain for cellular respiration. So just tell me the inputs are, the outputs are, the purpose is, and then where it happens. And then I want you to kind of tie this all back and compare it to photosynthesis, which is why I'm asking you to write about photosynthesis right after. Okay, so you're going to tell me about the light reactions, the Calvin cycle, and then if you want to, you can add the electron transport chain in. But I also want the inputs, outputs, purpose, and where for photosynthesis as well down here. So I'll be reading these when I grade your extra credit notes. Here is a breakdown of the diagram of what the parts of a mitochondria are called. It was on the first page, also simplified, obviously, because it's my hand drawing. But if you want more details, you can obviously copy this one down instead. But if you want the simplified version that honestly is all you really do have to know, you can also copy the one from the first page. Okay, so here's an actual free response that I want you to use. There is a diagram here of what happens when you break down pizza. And pizza contains proteins, it contains carbohydrates, and it contains fats. So I want you to write 3 to 4 sentences describing the fate of the energy-containing macromolecules in your pizza. So these 3 are the ones that contain energy. We know that nucleic acids like DNA do not, we do not break them down for energy, right? We just break them down and then use them to build more of our own DNA. However, these 3 are broken down for energy, and the table shows you what they're turning into, where they're used up. And so I want 3 to 4 sentences just describing what happens, and then you can also go into the details of cellular respiration after you run out of maybe describing what's happening actually to these 3 macromolecules. So kind of related to that, you can also use some of this information, but like I said, the sources of energy-rich molecules are carbohydrates, lipids, and proteins. And then vocabulary-wise, degrading reactions or reactions that are breaking down molecules are called catabolism, and they tend to be exergonic, so they tend to release energy. So when you break something, you're releasing energy. It's kind of like the analogy I made a long time ago with glue sticks. When you break it, you release light energy, for example, right? And the other way I remember this is that catabolism is breaking down because it's like cats, and cats tend to knock things off and break things. So catabolism is breaking for cats. Okay. Synthetic reactions or synthesizing reactions are anabolic reactions, and they build molecules. So they tend to be endergonic because in order to build something, you have to put in the energy to build that chemical bond, and therefore it's going to require or consume energy, and that's endergonic. Remember when we talked about Gibbs free energy? So the way that you can remember anabolic reactions is that anabolic steroids are steroids that build muscles, and they're the ones that bodybuilders get caught using to build their muscles to the extreme because they couldn't do it through a natural way, and then their muscles get huge and really, really big because they're just building these protein strands that weren't going to be naturally there in that size. So anabolic steroids are growing muscles. So anabolic reactions are growth reactions or building reactions. The last part that I'm going to talk about is related to the fact that there's not always oxygen present in a cell or in an environment that a cell is in. And sometimes cells are used to that. And sometimes cells thrive in environments like that. But there are also occasions where cells that do prefer to do the Krebs cycle are in an environment all of a sudden where they don't have enough oxygen to do it. And this results in these 2 different processes. So if oxygen is not available to the cell, fermentation or an anaerobic process occurs in the cytoplasm. So again, this is anaerobic, which means no oxygen. Aerobic means oxygen. Just think like aerobic, air, oxygen. Okay. So during fermentation, glucose is incompletely metabolized. It's not broken down all the way. So we're not getting the most efficient use out of it. We're not getting the most ATPs we can out of it. It's not even going to be occurring in the mitochondria. It's in the cytoplasm. Okay. Because the mitochondria part only works when you have oxygen. So you either produce lactate or you produce alcohol. And so lactate or lactic acid is in some bacteria, but mostly in fungi and humans. So for example, if you've ever done any sports or if you've ever run for a long time, you get sore. And that sore feeling or that burning feeling in your muscles is lactic acid. And again, the reason why it feels like that is because acids usually cause that burning kind of sour feeling. And that's what soreness is linked to. And so the reason why you feel like that is because your muscles are running out of oxygen. And so they are, instead of doing the reaction of producing a lot of ATP, because now there's no oxygen for them, they're going to be doing lactic acid fermentation instead of going through the Krebs cycle. And it's not efficient, but it's the best they can manage at the time. And that's why when you get tired, you slow down because your muscles don't have as much energy anymore. And then you also feel the burn. And this burn is lactic acid. There are other organisms like yeast that produce alcohol and carbon dioxide. And we use that. We use yeast in order to rise bread because the carbon dioxide bubbles cause the bread to rise and become nice and fluffy. And then when you bake it, the alcohol actually just bakes off because it evaporates at a pretty low temperature. So there are also many other chemicals that can be produced by various bacteria, but we generally just focus on alcohol fermentation and lactic acid fermentation. So why produce lactic acid or alcohol? So glycolysis produces NADH and pyruvate. And then it also produces 2 ATP, which is not a lot, but it's better than nothing. Okay. But at some point, you're going to run out of NAD+ because you're using it all up in glycolysis to make NADH, but it's not getting turned back because you're not doing the electron transport chain anymore because you're not doing the rest of cellular respiration. You're only doing glycolysis. So you have to regenerate NAD+ from somehow, because you need it to continue cycling even just for glycolysis. It has to accept the electrons. So what you do is you transfer the hydrogens to pyruvate. And when you transfer it, right, remember glucose is originally the 6 carbon, and glycolysis it's broken down into 2 pyruvates. You're going to add the hydrogen from NADH onto the pyruvates, and it's going to become either lactic acid or it's going to become alcohol. So you're going to have these 3 carbon alcohols or these 3 carbon acids floating around inside of the cells, and that's either going to be lactic acid or alcohol. Because then once there's no H attached to the NADH anymore, it becomes NAD+, it can go back, and you can continue doing more glycolysis at the bare minimum. Because doing glycolysis is better than not doing anything at all and not producing any energy. So it's just a kind of desperate attempt to produce some ATPs and continue a cycle. So the advantages of fermentation is that it works. It produces ATP. It's better than nothing. And it works in environments where there's no oxygen. So that could be when your muscles are tired and you're not breathing right when you're working out. It's still going to produce ATP, so you can still push through and finish your race or finish whatever you're doing. However, the disadvantages are that lactic acid and alcohol are actually toxic to your cells, right? So if you're consuming alcohol, you already know that it's bad for you, right? You learn in health class that it's bad for you. It's the same way that if your own cells are producing it, then it's also bad for your cells. It can degrade your cells. Lactic acid is also really bad for you in large quantities. That's why if you work out too hard or if you go too hard in a race, the next couple of days you'll be sore because your cells have to recover from having the lactic acid buildup so high in concentration. Okay, so it's bad for your cells. And then also the other disadvantage is that you're only, instead of producing 38 ATP, you're only producing 2 at a time. So you're dropping around the net difference of 36 ATP. Because 2 is better than nothing, right? So that your muscles keep working or your cells keep surviving and living because you have energy from ATP. But the difference is that you're really just working on like 2 instead of 38. And so it's not like a long-term solution, but it is a short temporary solution that does help you pull through. So you are going to go ahead and after you've done these notes, turn it in for extra credit, and then you're going to watch the Bozeman video on these notes and then turn in that video guide as well. Don't forget to work on your chapter 8 summary and learn smart, which is on cellular respiration, because that is due on Friday.